Hi, and welcome to the second part of the cerebrum. Here we are going to talk about the white matter. The white matter is formed by fibers. And there are three types of fibers: commissural, association, and projection. Commissural fibers are fibers that communicate both cerebral hemispheres. And there are three: commissure of the fornix, Roztral Commissure, and the Corpus Callosum. The commissure of the fornix communicates both fornices, so it belongs to the archicortex, right? You have it here in this drawing. These are the fornix, parts of the fornix, and then the commissure of the fornix. The rostral commissure is paleocortex, communicates the olfactory bulbs and also the piriform lobes. And finally, here is in green, and finally the corpus callosum. The corpus callosum communicates the neocortices, right, between both hemispheres. And you have it here, the corpus callosum, and here the corpus callosum. Association fibers are fibers that communicate different areas, but inside of one cerebral hemisphere. They do not cross, or they do not project to different areas apart from the cerebrum, right? The projection fibers are fibers that project outside the cerebrum, or they get to the cerebrum. And here we have the internal capsule. We begin by talking about the corpus callosum. Corpus callosum comes from the Latin meaning hard body. The corpus callosum communicates the neocortices of both cerebral hemispheres. Here is a dorsal view. I have removed the parts of the gyri that are close to the longitudinal fissure, the marginal gyrus and the ectomarginal gyrus of both cerebral hemispheres. And we see the fibers that communicate the corpus callosum. Again, the corpus callosum between both cerebral hemispheres under the cingulate gyrus. Here is a Muligan staining, and we identify the corpus callosum. The corpus callosum has different parts. One is the rostrum. The rostrum communicates motor cortices, primary and supplementary motor cortices. Then we have the genu that communicates the prefrontal cortex between both hemispheres. Then we have an extended body of the corpus callosum that communicates motor and some aesthetic cortex. And then finally we have the splenium that communicates temporal and occipital cortices of both hemispheres. This is a dorsal section of both hemispheres, cerebral hemispheres. You see the coded nucleus with a long tail, right? The head, the body, and the tail. And here we have corpus callosum, right? The rostral commissure. The rostral commissure, as we have explained before, communicates the olfactory bulbs. Where is this commissure located? Here. It is located just rostral to the columns of the fornix, right here. Right here is the rostral commissure. You see the rostral commissure, and the rostral commissure is located at the same level, but dorsally, to the optic chiasm, because both are originated from the lamina terminalis that extends in the embryo, the rostral end of the diencephalic vesicle. And as you see here, this is the rostral commissure, and behind we have the columns of the fornix. Okay? So remember that the rostral commissure just dorsal to the optic chiasm. Let's have a video. Now we split both cerebral hemispheres because I have cut the corpus callosum. And this is just to identify here the rostral commissure. This is the rostral commissure here. Right? It's quite easy to identify in a dissection, the rostral commissure. Okay, so now we go to the fornix. The fornix, that means from Latin volt. Okay, as we have said previously, they are the fibers that are leaving the hippocampus and they form a volt around the thalamus. So it's formed. So the fornix is formed by commissural and association fibers. Association fibers are fibers that communicate different structures inside one hemisphere. They do not cross to the other side. So these are fibers that are coming from one hippocampus. They go up, they go rostral, they form the body, and then the columns of the fornix that reach the mammillary bodies of the hypothalamus. However, there are some fibers that communicate both crucifornis, and these are the commissural fibers. So here you see the corpus callosum and the fibers of the columns of the fornix going rostral and down in order to reach the mammillary bodies, passing just caudal to the rostral commissure. There you have that, another dissection where you see the crucifornis going up and being related with the telencephalic septum here. Okay. Now this is a dorsal view of an encephalon. It's covered by the meninges. The cerebellum behind, the cisterna magna. And underneath we identify the hypophysis. Okay, I have removed the meninges. This is the longitudinal feature, right? Okay. All right. With all the gyri, the occipital cortex, the cerebellum, and the cisterna magna there. You see, this is the cisterna magna. This encephalon has no dura, but has arachnoid. So this we have the hypophysis and the optic nerves with the optic chiasm, and the internal carotid arteries. You see the olfactory bulb here, and continues caudally as the olfactory peduncle, and this sulcus is the lateral rhinal sulcus that separates the neocortex from the basal portion of the paleocortex. And what I want you to show here is here you see this is the piriform lobe, is entorhinal cortex, continues with the parahippocampal gyrus. This is entorhinal cortex. This is the entorhinal cortex. Now we separate both cerebral hemispheres and we identify the corpus callosum. Well, right? With the rostrum, the body, and the splenium. And on top, the cingulate gyrus. See, this is the corpus callosum. And splenium of the corpus callosum. Now we remove part of the neocortex in order to identify here is the insula, the insular cortex. This is the piriform lobe that extends caudally as the parahippocampal gyrus, the white matter. the insular cortex or insula. Having removed most of the neocortex, we have the transition cortex, that is the cingulate gyrus, on top of the corpus callosum. Right. We remove the cingulate gyrus and we identify here the corpus callosum. Commissural fibers. Okay, as you see, the cingulate gyrus continues caudally as the parahippocampal gyrus and continues as the piriform lobe. And this portion, the caudal portion of the piriform lobe and the parahippocampal gyrus, are known as the entorhinal cortex. Fibers from the entorhinal cortex reach the hippocampal formation. Can you see the corpus callosum there? And the amygdaloid body. This is the amygdaloid body inside the piriform lobe. This is the piriform lobe, right? So now we identify the fornix. This is the fornix, the corpus callosum, and underneath here we have the telencephalic septum with the pellucid and cellular portions. Telencephalic septum, you see how it is closely related with the fornix. Right. This is a dorsal section in order to now to focus on the internal capsule. Okay, the cerebellum behind, right? The coded nucleus right here. Okay, the hippocampal formation, the parahippocampal gyrus here, parahippocampal gyrus, both thalami. And then you see this is the internal capsule. The internal capsule is divided in three portions. The rostral crus here, the genu and the caudal crus. The rostral crus is between the coded nucleus and the lenticular nucleus. The caudal crus is between the thalamus and the lenticular nucleus. This is the lenticular nucleus. And the genu is in between. So the rostral and the caudal. And in the caudal crus, we have the optic radiation. Let's have a look at the optic radiation. In this dissection, I have removed part of the neocortex in order to identify this, the optic chiasm with the optic nerves, the optic tract, fibers that are coming from the optic chiasm until the lateral geniculate body of the thalamus. And fibers from this lateral geniculate body, they leave and they form the optic radiation that projects to the occipital cortex. Let's have a look. You see the optic tract here, the crus cerebri, right? And the optic radiation. This is the optic radiation. And you see the lateral geniculate body here. And these are optic fibers, right? So remember, this is the optic radiation that is part of the caudal crus of the internal capsule. So if we make a transverse section of the encephalon, here is a Muligan staining. And what do we have? And we are going to focus on the white matter. We see the corpus callosum, these commissural fibers, the Roztral Commissure, commissural fibers, ¿vale? Fornix association fibers, right? The internal capsule, projection fibers. And when the corpus callosum that is formed by commissural fibers meets the internal capsule, then we have the centrum semiovale. Fibers from this centrum semiovale, some of the fibers may project to the cortex. They form the corona radiata. However, we also have fibers that are longitudinally. So fibers that connect different areas, association fibers. Some of them perpendicular, some of them longitudinal. So what are they? These are the external capsule and the extreme capsule. The external capsule is between the putamen and the claustrum, and the extreme capsule is between cortex and claustrum. And these are associative fibers. Also, there is this subcallosal fasciculus here located, okay, because it's located underneath the corpus callosum, subcallosal fasciculus. And this is a fasciculus that connects frontal cortex with occipital cortex. We also have here at this level the superior longitudinal fasciculus. It's a fasciculus formed by associative fibers that connect the frontal cortex with the parietal cortex with the occipital cortex. We also have the uncinate fascicle. This fasciculus connects the temporal cortex with the frontal cortex, or the inferior longitudinal fasciculus that connects the occipital cortex with the temporal cortex. In these enlarged images, this is a Bielchowski or Muligan. This is an impregnation, and this is a staining, right? So we have the coated nucleus, the internal capsule with the pallidum and putamen and claustrum, internal capsule with the putamen and claustrum, right? And this number two here is the superior longitudinal fasciculus. This fasciculus is located just on top of the putamen, intermingled with the, or mixed with the fibers of the internal capsule. And here, just very medial to the centrum semiovale and at the dorsal to the most lateral portion of the coated nucleus is the subcallosal fasciculus. Is a fronto-occipital fasciculus, right? Because here we have the corpus callosum. Okay? So this fasciculus is located just top of the lateral aspect of the coated nucleus. This is the tail of the coated nucleus. So now we are performing a dissection with the technique of fiber dissection. We remove part of the neocortex, the sylvian gyrus, suprasylvian gyrus, and we identify all these fibers that form the internal capsule. Okay, we keep removing cortex and white matter. And we discover the superior longitudinal fasciculus here, these fibers that connect the occipital cortex with the frontal cortex. We keep removing cortex with internal capsule. And we reach the centrum semiovale here. You see all these fibers of the superior longitudinal fasciculus. Remember that the centrum semiovale is when the internal capsule meets the corpus callosum. It's this area here. Okay. We keep dissecting with this technique. In order to achieve these images of the fibers, the encephalons have to be frozen and defrozen several times. So you see, these fibers here belong to olfactory tracts. These fibers belong to the fornix, and these fibers belong to the optic tract. And then we have the centrum semiovale. You see, the optic tract coming from the optic chiasm and going up to the lateral geniculate body. You see the corpus callosum here that joins the internal capsule and forms the centrum semiovale. It's this area. Now we discover the coded nucleus here. This is the coded nucleus, and we introduce the forceps in the lateral ventricle. The centrum semiovale again. And now we have cut through the centrum semiovale, and we identify the lateral ventricle here. You see, this is the coded nucleus, the corpus callosum, and we see the lateral ventricle. This is the lateral ventricle here. Now we proceed the fiber dissection, and we go to the hemisphere on the right, but we are going from medial to lateral, right? We identify here the coded nucleus, right? This is the coded nucleus. This is a medial vision with the tail of the coded nucleus and the head. The lateral ventricle here. So the coded nucleus, you see, is gray matter. We remove part of the coded nucleus and we identify what is lateral to the coded nucleus, the internal capsule. This is the tail of the coded nucleus. And lateral, lateral to the coded nucleus, we have the putamen, the lenticular nucleus. And here we also have the lenticular, the pallidum. So you see the lenticular nucleus is lateral to the internal capsule. These are fibers from the internal capsule. This is another dissection. This is a dorsal view with the longitudinal fissure. And we split the encephalon through the midplane. And we identify the corpus callosum, right? The interthalamic adhesion, mesencephalon, pons, medulla oblongata, cerebellum, right? Cerebral hemisphere, right? The fornix, the telencephalic septum, with the pellucid portion and the cellular portion, paraterminal gyrus. Okay, you see the pellucid portion here and the cellular portion of the telencephalic septum. We remove the gyri from medial to lateral now again. The corpus callosum here. And now we have removed the cingulate gyrus with the part of the parahippocampal gyrus. And as you see, these fibers are fibers from the corpus callosum, and these are fibers from the corona radiata. Okay. We have removed the splenial gyrus. And we identify here the dentate gyrus. This is the mesencephalic aqueduct and mesencephalon. From a caudal view, the mesencephalic aqueduct here, the fourth ventricle, and the parahippocampal gyrus has been removed. Now the brainstem has been cut transversally at the level of the mesencephalon pons. Okay. As you see, the parahippocampal gyrus was continuous with the piriform lobe. Now the thalamus has been removed. And what do we see from medial to lateral, from the inside? The splenium of the corpus callosum, right? And here we see the hippocampal formation. Together with the fornix. This is part of the fornix here. Okay, this is part of the fornix. And more externally, forming part of the lateral ventricle, the walls of the lateral ventricle, the hippocampus. Now we push the hippocampus out. The hippocampus, right? And this is the lateral ventricle. You see how the lateral ventricle has a C shape as the hippocampus and the fornix. Here again. And when we remove the hippocampus and fornix, we clearly identify the lateral ventricle. And another wall of the lateral ventricle is the caudate nucleus with its tail. So you see the caudate nucleus here and the tail of the caudate nucleus. So if we compare the anatomy of our domestic animals, the dog, cat, you see the cat is smaller. We have seen it before. The pig with the long cruciate sulcus is directed rostrally, not as in the case of a dog. Or in the sheep with a short cruciate sulcus. Or in the horse. The horse has an ovoid shape and the cruciate sulcus is only visible in the immediate plane. Or in the ox, pigmented. So in the sheep is also pigmented. Well, thank you very much.